Ladies and gentlemen, welcome to Compass here live on Quotidiano TV. Today with a very special guest from San Francisco with Professor Michael Geschwind. Professor, welcome to Compass. How are you?
Thank you. Great to be here and excited to talk to your diverse audience.
Professor, ladies and gentlemen, if this was an interview designated to neurologists only, I wouldn't have to introduce Professor Geschwind, since he's one of the world's most famous neurologists, an expert in aging and prion research. Ladies and gentlemen, let me tell you, since not all of you are neurologists, some are physicians, some are other sort of scientists, Professor Geschwind is a professor at the University of California, San Francisco. You received, Professor, your MD and PhD degrees in neuroscience through the National Institute of Health, sponsored medical scientist training program at the Albert Einstein College of Medicine in New York. You completed your internship in internal medicine at the University of California, Los Angeles Medical Center, and your neurology residence at the Johns Hopkins University School of Medicine in Baltimore, and your fellowship in behavioral neurology at the University of California in San Francisco. And currently you are a professor of neurology at the Memory and Aging Center. Professor, it's a real honor to host in my today's talk show. Related to prions, let's start from the bottom. Prions are actually the source, this proteinaceous infectious agent for the spongiform brain. Where do you think this name prion comes from?
Well, actually, we know where the name comes from. When Dr. Prusiner was trying to describe how a protein that could change shape and thereby by misfolding cause other similar proteins to misfold and result in disease, and that this protein was identified in infectious and sporadic diseases, he thought of the protein as a proteinaceous infectious material. And he took the letters prion from that term proteinaceous infectious. And he thought that ion sounded better than oin. And so he calls it a prion, prion. And it's interesting, my British colleagues and those who train in British English call it a prion. Dr. Prusiner calls it a prion. Since he named it, I think he's right. But then again, there's always a difference in pronunciation between American and British English. So I think both are acceptable. But yeah, so I call it a prion, but other people half the people at prion meetings call it a prion, half call it a prion. But it stands for proteinaceous infectious particle. And it's a little bit of a misnomer because some people think, oh, it's always an infectious disease. But that's not necessarily true. It's infectious in that it's transmissible from cell to cell, but not every form of prion disease has been shown to be transmissible from person to person. So not all forms occur via an infectious manner. For example, in human prion disease, 85 to 90% are spontaneous disease, a spontaneous misfolding of a normal prion protein into the misshapen form, the prion. And then about 15% are genetic, and less than 1% of all human prion diseases are known to be infectious or transmitted, or I like the term acquired. Even that term prion has to be, and its derivation has to be interpreted cautiously, because you don't want to always assume that every form of the disease has occurred because of an infection. So when I diagnose my patients with prion disease, they always want to know, well, how did I get that infection? Where did I catch it from? And what I answer them is, well, it's you in most cases, you get it spontaneously, just like you get most cases of Alzheimer's, most cases of Parkinson's disease, they occur spontaneously. There might be genetic or environmental risk factors, but for the most that influence one's risk, but for the most part, it's spontaneous. And sorry for that long answer.
No, it's fine. I was listening carefully, as much as for sure the audience was also listening carefully. But I mean, these prions, they induce this type of spongiform brain, basically. During the pathogenesis of prion disease, which affects the brain, we'll talk about the immune aspects of prion disease. We also have this brain damage with holes, with vacuoles produced within the brain, if I'm not mistaken.
You say that last part? Yeah.
So the spongiform brain, I mean, it's inducing severe damages to the brain, the prion disease.
Yes, exactly. So in all cases, there's damage to the brain. The damage to the brain is variable. There are a couple of pathological hallmarks. There's neuronal cell degeneration. There is proliferation of supportive cells like gliosis. Those are the same types of cells if you had a stroke or a brain injury. After the debris is cleared, then other cells, astrocytes, come in to kind of clean up the mess, and they leave a scar. It's almost like scar tissue. And then a third is what pathologists actually tend to call vacuolation. The old terminology is spongiform change, and it's still used, but and we call it spongiform because if you cut brain tissue and you look at it at a very high level, but under the microscope, you see holes in the tissue almost like a sponge. It turns out that they aren't really holes. They are spheres, fluid-filled vesicles within the arborization of nerve cells. Think of a nerve cell like a tree. You have the branches of the tree, you have the roots, you have the body of the tree, which are maybe near the top of the tree. And all those branches are what we call dendrites. And those vacuoles are fluid-filled spherical vesicles that fill up. And in prion disease, and most diseases where those occur, it's abnormal. And so you get the vacuolation or spongiform change. Interestingly, not all prion diseases have spongiform change or vacuolation, which is why I don't use the old term transmissible spongiform encephalopathy. And then also, not all have been proven to be transmissible. So there's this old term transmissible spongiform encephalopathy. And I tend to just use the term prion disease because not all prion diseases fit known transmissible, not all fit spongiform.
But do you think, Professor, that under certain circumstances, the term might be at breach with at least current ethical considerations? Prion. In the etymology of the word, although Prusiner says that he was thinking of EDIT-PF in an interview with Professor Lanka a couple of years ago, the prion was Hippocrates' trepanation tool, the so-called modulus. The name Hippocrates gave to his crown drill bit was a prion. I mean, we are not talking about a coincidence that a prion would mean in Greece cheese or milk or wheel. It's a trepanation tool. It's a crown drill bit. And a trepanation tool is an instrument, let's say a neurosurgeon might be dealing with. But I mean, it's an instrument of a physician. Every physician is making an oath on. So it's quite surprising to me, the coincidence that a pathogen agent inducing vacuoles in the brain has the same name as Hippocrates' trepanation tool.
Interesting.
I've asked Professor Prusiner, invited him twice in interviews related to the etymology of his name and how he refers to the acronym, which you mentioned at the beginning of our interview, and how he in the interview with Lanka refers to EDIT-PF. And I've asked him if he would agree on talking about the new term, because in my personal point of view, because of my education and my moral conduct and my ethical values, in case I was suffering, I would have to suffer for prion disease. I wouldn't like the name of the disease to be synonymous with the trepanation tool. I do not want a physician to tell me literally that, Mr. Golban, you have a hole in your head. And I mean, Prusiner received his Nobel Prize in 1997, but we're not talking about any physician in this world. We're talking about Hippocrates' trepanation tool. And I will interview next week 2 professors from the Paleopathology Institute of Pisa who have published several papers on the prion. It was Hippocrates modulus. So it's not any drill, drilling a hole in the skull, it's a crown drill bit. So I believe Prusiner doesn't speak Greek, so he has really he's not guilty on that, and I do not accuse him of having chosen this definition premeditately, but ethical values prevail much higher than one's personal knowledge in the history of medicine. And once this is known, I think from an ethical perspective, it should be incompatible of, I mean, imagine tomorrow, after our discussion, you talk to a patient, tell him you have a prion disease. You'll keep in your mind that this guy has a hole in his skull because Hippocrates' trepanation tool was a prion.
Yeah, I mean, I understand the connection you're making. I just don't think most physicians current day would be referring to the, to that tool. I'm ignorant of that. I've been in medicine for about 30 years and I'm not aware of that. So thank you for teaching me that point.
My pleasure. I don't my intention on teaching you.
No
ethics is something we all ought to put the emphasis on. I mean, particularly because since COVID, prion disease, I think, are a real issue. COVID is associated to prion disease as well, if I'm not mistaken. And if you could please tell us something why COVID might be associated to prion disease.
Yeah, there have been reports about increased incidence of prion disease with COVID, and I would not be calling at this point COVID a prion disease. We can talk more about, I think we might have time more to talk about COVID, but anytime there's an outbreak or something like COVID, obviously there's been little in our lifetime that's been like COVID since the Spanish flu, but whenever there have been other outbreaks or whatnot, I can't tell you how many contacts I get from patients and families asking me, they got COVID and then they got prion disease, or they got the vaccine and then they got prion disease. And, given the vast number of people who've gotten both, you're going to expect people to have both, to have COVID, either the vaccine or have had COVID itself and get prion disease. And so until you get the summary data statistics and we look at the incidence of COVID during this period, it will be hard to know. I can certainly say that I had far fewer referrals for prion disease during the pandemic than before. And you might mistakenly say, oh, COVID or the vaccine protects against prion disease. But no, it's just that people weren't being referred because of the pandemic and medical isolation and that type of thing. So I think cause and effect is difficult. In terms of the idea of what is a prion disease, I probably would limit it. And if you asked us 10 years ago, we would say there's essentially one class of human prion diseases. They deal with the prion protein, the prion protein, PRP, in humans and in non-human species. And it's a misfolding of the normal shaped prion protein, the role of which is still a little bit controversial. It has some role in possibly neuroprotection, in synaptic plasticity, possibly in memory formation. But you can remove a prion protein genetically from a mouse, embryologically or even before, and they live a normal lifespan. But if you do detailed experiments and studies on the mice, you find out that they have some mild sensory neuropathy, some sensory problems. We know that there have been people born with only one copy of the prion protein gene, and presumably they live a normal life. And so we think even humans can have a reduced level of the prion protein. We don't know if they can survive without it, but probably they can, as a mice can, but we're not sure.
But what sort of behavioral changes, sorry for interrupting you, Professor, what sort of behavioral changes do appear? I think I've written a paper written by Professor Guzzi, and he also refers to that these mice tend to be more aggressive. I don't know if I kept it properly in mind or not, that mice with this knockout gene with this knockout on the prion gene. Do they have a normal behavior, or they tend to be more aggressive?
Well, to my knowledge, they have normal behavior, but I'm not familiar. I'm sure I've read almost all of Guzzi's papers on his knockout. He's done some of the most beautiful work, where one of the problems with a lot of the knockout models was that they weren't very clean models, in that they not only knocked out the prion protein gene, but sometimes they knocked out either a little bit at either end of the gene. And those changes alone that were not in the gene were actually the cause of the phenomena that was seen from the knockout. And so Dr. Guzzi did some very nice experiments in which he made clean models, and he showed that some of the changes that people had reported in their messier models did not appear. I don't recall the behavior. There are behaviors that Giovanni Malucci has shown in terms of mice. They have certain kind of nesting behavior. And if you knock out or restore the prion protein, you can restore that nesting behavior. I'm not familiar within humans reducing the gene that there's any behavior known. And there have only been a few cases in which we know that people have only one copy of the prion gene, and the assumption is that they have half or approximately half the normal level. These are from large genetic databases, so those patients haven't been really well characterized. Some of the treatment trials that we're about to embark upon, their goal is to reduce the normal prion protein, the substrate. So in prion disease, you have your normal prion protein, and it comes in contact with a misfolded protein that Stan Prusiner called the prion. That causes the normal prion protein to misfold. Now you have two misfolded disease-causing forms. They come in contact with two normal forms, convert them. Now you have four. It's an exponential process. And the idea in some therapies is to prevent those two from coming together, so maybe a small molecule or an antibody that prevents binding, or perhaps just removing the normal protein, reducing it. And if you reduce the normal protein, the transformation doesn't occur. You have a few misfolded proteins, the prions, and you don't have that curve, the exponential curve going up. So that's one of the mechanisms. And our hope is that will not cause harm to patients. Not only do we want it to help patients by reducing the risk of prion disease, particularly in a person who has a genetic mutation causing it, or maybe stopping or slowing or reversing the disease in a patient who already has symptoms, be it from a genetic, sporadic, or acquired cause. And we hope is that there won't be any negative effects like what you're alluding to.
What about the success of the anti-prion vaccination? I mean, it's quite interesting and not surprisingly at all, particularly the mechanism of the active anti-prion vaccination. It's something which has some aspects resembling the anti-COVID vaccination. I mean, we have a protein antigen, or there's a fabulous paper written by Konstantinos Xantopoulos and Dimitra Dafo, but there are also other papers starting with 2003 describing the various advantages and certainly also disadvantages of the passive and active anti-prion vaccination. And we have the plasmid in the active vaccination, the plasmid, the RNA, the DNA vector vaccination, much similar to other vaccines with a protein antigen, which we have also experienced during COVID. Do you think that this sound and thorough research in the area of prions and anti-prion vaccination was for the benefit of us all, particularly during this severe pandemic, having easier facilitated us vaccines, so much needed vaccines against COVID?
Let me see if I could, if I understand your question. Is the question about the benefit? Well, let me answer this question. The benefits and comparing an active versus a passive vaccine.
No. If the current vaccine, sorry, if my question was confusing, if the vaccines which were administered during COVID also benefited from the research of the active anti-prion vaccination, if the success in the anti-prion vaccination research have facilitated vaccines against the so much needed vaccines against COVID.
I wasn't aware of any mRNA type vaccines being used in the against prions in the pre-COVID area.
Well, 2016, Immunobiology of Prion Disease, a paper written by Konstantinos Xantopoulos, Dimitra Dafo, and Theodoros, I cannot read these Greek names, I apologize. And there they describe the active and the passive immunization. And among the active immunization, you have the mRNA type of F vaccine, where with the peptide-based active vaccination, you have several examples, and then you have the DNA vaccines, and then you have the immunization with the PRP displaying viral construct. Well, but we should not go into details in this question. I thought that benefits from the anti-prion vaccination would have facilitated the so much needed vaccines against COVID.
It's possible, I suppose. Yeah, I wasn't aware that they actually had created any mRNA vaccines, but it was just one of the many ways in which you could create a vaccine, and why not do it for prion disease like you would for COVID?
Why not? Why not? I mean, we are all happy if human beings really have a benefit from scientific research, particularly in such a sophisticated and complex matter as the prion research.
Yeah.
One more, since we're approaching the end of this very interesting interview, you said that you have particularly recently encountered less cases of prion disease than in the past. What about autoimmune encephalitis? Have you there maybe encountered more cases than in the past?
Yeah, no, so I also see a lot of patients with autoimmune encephalitis, and usually these are patients who have syndromes due to autoantibodies that attack different aspects of the central nervous system. A classic example might be anti-LGI1 antibody or anti-NMDA receptor antibody. And most of these are against antibodies that are auto-generated against synaptic proteins, or the connections between nerve cells, or maybe proteins that are on the cell bodies of nerve cells. And, we had a reduction during COVID, we had a reduction in referrals of all types. So autoimmune, prion disease.
We have them all.
Because of how the medical system shut down, and we weren't even accepting patients in the hospital unless they had COVID, essentially. So, certainly I'm beginning to see an increase, a normalization of the referrals for these syndromes. And in fact, we're able to see more of them in some ways because of the of telemedicine. Now, during COVID, we were able to begin to see patients via telemedicine by Zoom much more readily. We could do it in the past, but it was hard to bill for. Insurance didn't like it, Medicare, and at least in the US now, it's, and hopefully it will stay to this extent, where I'm able to see patients who normally would have to drive 6 or 7 hours to see me, and now I can see them, or fly across the country, or even from India, and now we can see them by Zoom.
Much
One of
like
the benefits of the pandemic,
much like our audience can see you by Zoom today in Romania, Professor.
Great, thank you.
It was such a joy and talking to you today, and I really feel deeply honored having answered my questions. My best regards to San Francisco, and looking forward to seeing you soon, Professor.
Okay, thank you very much.
Thank you very much for having joined us, and all the best. Have a nice afternoon. Bye-bye.
Bye-bye.